Vande Gujarat.
Good morning, all of you. All the NET CSIR aspirants, I take an opportunity to discuss something about the few questions and a brief outlook on structural organization and function of intracellular organelles. I am myself Dr. Akshaya Gupte, Head Department of Microbiology at Natubhai V. Patel College of Pure and Applied Sciences, Vallabh, Vidyanagar. First of all, we will talk about what are cells. All organisms are very complex, made up of several building blocks, say for example carbohydrates, proteins, lipids, and so on. And so these all organisms are complex, and all the cells, whether a prokaryotic cell, whether a eukaryotic cell, all these cells, they all are derived from by the process of cell division. And this cell division takes place from the pre-existing cells. Now when we are talking about the cellular organization of the cells, living things, they all are constructed of the cells, and living things may be a unicellular, say for example your bacterial cells, or a multicellular, maybe what you can say any animal cell or a plant cell, it is a multicellular cell. Now these cell structure, they are diverse, but all cells, they share a common characteristics. Now these cells which are there, if we divide the cells, we can divide the cells into two types. One is the eukaryotic cell and the other is a prokaryotic cell. Now further, if we talk about the division of the prokaryotic cell, then it is clearly said to be a eubacteria and secondly an archaebacteria. Eubacteria are the true bacteria, they are the bacteria which we are classifying into say different organisms like your E. coli, Bacillus species and so on. Whereas the archaebacteria, they are the old or the primitive bacteria which are there. Whereas the eukaryotic cells or the eukaryotes which are there, they appeared almost 1.5 billion years ago on the earth. Now there is only one kingdom, that is the eukarya, and this eukarya has got the presence of the protists, it has got the presence of the fungi, animals as well as the plantae. Now the cell theory, when further it was during the earlier period of the evolution, during the study of the cell division, the cell theory was formulated, and the cell theory states that all organisms, they are composed of one or more cells. Secondly, cells are the basic unit structure and functions in all the organisms, and all cells come only from the pre-existing or the other cells. This cell theory was given way back in early 1800s, wherein all the organisms, they can be put into this. Now if we see the size of the living things, it is ranging from 0.1 nanometers to 1 kilometer. See, with the help of the electron microscope, we are able to see atoms, amino acids, then proteins, viruses, most bacteria, plant and animals, cells and the human eggs. Whereas from the light microscope, we can always see from 1 micron to almost to 1 millimeter. And with the help of human eye, from 1 millimeter to 100 meters. So we are seeing the different organisms of the life. We are seeing the different living things as small as atoms to as big as a blue whale. Now these all organisms, these all organisms which are there, they have appeared with the help of the process of cell division. Now when we are talking about the eukaryotic cells have a well-developed nucleus and which controls the working of all the cell. All the cells of the eukaryotes, they are surrounded by a plasma membrane which is made up of phospholipids and the protein molecules. Now, that means the nucleus is a very important organelle among the eukaryotic cell, which is controlling the working of the complete cell. That means we can say that this nucleus, the presence of the nucleus, is the workhorse of the complete, or it is a ringmaster which directs all the other organelles to function in some way or the other. Now, this is a basic structure of a plasma membrane, which we are seeing a phospholipid bilayer which is present. There is a presence of the protein molecules and there is a lipid molecules also. So there is a presence of the hydrophobic as well as the hydrophilic ends which are there, and that makes the complete plasma membrane. So this plasma membrane, what is the function of this plasma membrane actually? The plasma membrane, it regulates whatever enters it and exits the cell. So the plasma membrane regulates its entry and exit of the nutrients, or you can say of any molecule within the cell. Inside the plasma membrane, the nucleus is surrounded by a watery fluid, that is the cytoplasm. Plants have a cell wall in addition to the plasma membrane, whereas the animal cells which are there, they lack the cell wall. There is only the presence of the plasma membrane which is there. So animals and plant cells have different organelles. These organelles which are there, they are compartmentalized into various compartments of the functions within the cells. Organelles of the animal cell as well as the plant cell, they are similar to each other, except the presence of the centriole are present only in the animal cells, and the chloroplast is present only in the plant cells. So there is a difference existing between a plant as well as an animal cell, in which we are able to see the compartmentalization of the cell. If you look at the compartmentalization of the cell, then one should remember the first time a cell which was described as a unit of life by Robert Hooke. He saw a cork which was compartmentalized just like the hive of the bees. So it was just like the beehives, he just saw the cork compartmentalization in the microscope. So the organelles of the animals as well as the plant cells, they are quite similar to each other, except the presence of the centrioles that are present in the animal cell and the chloroplasts which are present only in the plant cells. Now if you look at the animal cell anatomy, if you observe the presence of this animal cell, you are able to observe the presence of the nuclear pore, you are able to observe the presence of the chromatin, nucleolus, nuclear envelope, that all constitutes one organelle, that is the nucleus. Then that means a nucleus is not a simple organelle. It does contain a large number of compartmentalization. Also again, it has got a presence of the nuclear pore, chromatin, nucleolus and the nuclear envelope. If we talk about the endoplasmic reticulum, then it is having two types of endoplasmic reticulum parts, you can say. One is the smooth endoplasmic reticulum and the other is a rough endoplasmic reticulum. There is a presence of the peroxisomes, there is a presence of the cytoplasm, there is a presence of the ribosomes, which are supposed to be the site for the synthesis of our very important biomolecules, that is your proteins, that is your enzymes, which are being synthesized within the ribosomes. There is the presence of the Golgi apparatus, vesicles, plasma membrane, then the presence of the cytoskeleton, which is very, very important in the animal cell, which is having the actin filament, intermediate filaments, the microtubules, and so on. There is also the presence of the lysosomes, which are said to be suicidal bags, as well as the mitochondrion, which is the powerhouse of the cell, which generates the ATP for all the animal cells, through which the various functions of the cell can be carried out. So along with it, there is also a presence of the centrioles, there is a presence of the polyribosomes, there is a presence of the actin filaments, which you all are observing on this. Now this particularly, my presentation today, first of all, I would be discussing completely in theoretical way the overview of the organization of the cell. Thereafter, in the second part, I will deal about 30 to 40 questions which have appeared in certain NET examinations or in IISc or in other gate examinations too. So first of all, we are going further into the details of the different organelles of the cell. Now further on, if we see the presence of a plant cell anatomy, and if you compare the two, there is also in the nucleus, it is also the presence is quite similar. There is a presence of a nuclear pore, there is a presence of a chromatin, there is a presence of a nucleolus, there is a presence of a nuclear envelope. They all constitute within the nucleus. There is also the presence of the rough endoplasmic reticulum, smooth endoplasmic reticulum, but there is also a presence of one more thing, that is the cell wall. Now, apart from the cell wall, it is an outermost covering which provides rigidity, which provides shape to the cell. Along with the plasma membrane, we are observing the presence of the cytoplasm, we are observing the Golgi apparatus, there is a presence of the middle lamella, intracellular spaces, cell wall of adjacent cells, mitochondrion, actin filament, ribosomes, chloroplast, central vacuole, which is important in the plant cell, as well as the microtubules. Now we come to the structure of the nucleus. It has got the chromatin, nucleolus, nuclear envelope, and the nucleoplasm. So chromatin, there is a presence of DNA and proteins. In nucleolus, chromatin and ribosomal subunits. Nuclear envelope, double membrane with pores, and nucleoplasm, it is a semi-fluid medium which is present inside the nucleus. This is the structure of the nucleus and the nuclear envelope. On the right side, if you are observing the electron micrograph of the nuclear envelope, which are showing the presence of the pores. Now you are seeing the nuclear pore, inner membrane, outer membrane, as well as the nucleolus and the chromatin. If you see it in detail, there is the section which has been shown, which shows the outer membrane, which shows the nuclear pores in a bigger manner. There is an inner membrane also. Nucleus and nucleolus. So when we talk about the general definition of a nucleus, nucleus is the genetic control center of all the eukaryotic cells. In most cells, there is only one nucleus. It is spherical and most prominent part of the cell, and it makes up almost up to 10% of the total cell's volume. So this may be one of the simplest questions asked, is that what constitutes, how much amount constitutes the total cell volume is within the animal cell. So answer, it can range from, certain books say 10%, certain books say 10 to 12% can be said. So the correct answer, it can lie between 10 to 12% of the total cell volume of the eukaryotic cell is taken up by the nucleus. It has a unique structure as well as a function that is very much essential to the cell. So when we talk about the history of the nucleus, it was discovered in 1831 by a Scottish botanist, Robert Brown. He suggested that the nucleus played a key role in fertilization and development of the embryo in the plants. The name nucleus was further derived from a Latin word for kernel or the nut. Main characteristics of the nucleus. It is a membrane enclosed organelle found in the eukaryotic cells, generally found in the central region of the cells in the animal cells, roughly spherical shaped, what we said earlier also, and largest and the most easily seen organelle in the eukaryotic cell. On the left, if you are seeing the picture, the three-dimensional picture which is showing the nucleolus, the nuclear pore, chromatin, as well as the nuclear envelope. What are the primary functions of this important organelles within the cell? It is considered to be a repository of genetic information of DNA as well as the RNA. It enables the synthesis of nearly all the proteins, houses the nucleolus, responsible for the production of ribosomes, selective transportation of regulatory factors and energy molecules through the presence of the nuclear pores. So it does have got a good important primary functions. The first and the foremost being your repository of genetic information, whether it is a DNA, that is deoxyribose nucleic acid, or the RNA, that is the ribose nucleic acid. We have already seen the structure quite in detail, what it contains: nucleolus, nuclear spore, nuclear envelope, and so on. So what is this nuclear envelope consist of? If we see the picture on the right, we are seeing the presence of ribosomes, we are seeing the pore complex, we are seeing the close-up of the nuclear envelope completely, outer membrane, inner membrane, as well as the nuclear lamina. So it consists of the phospholipid bilayer membrane, it consists of the nuclear pores, as well as it consists of the ribosomes. This nuclear envelope which is there, it is also a double-layered membrane perforated with pores which controls the flow of material which goes inside and outside of the nucleus. So it is a barrier which is present within the nucleus, that is the nuclear envelope. It is a double-layered membrane perforations with the specific pores which will control the entry and exit of the molecules within inside and outside of the nucleus. The outer layer of this nuclear envelope, it is connected with the endoplasmic reticulum, thereby communicating with the cytoplasm of the cells. The exchange of the large molecules, that is the proteins as well as the RNA, between the nucleus and the cytoplasm happens at this particular place, that is the nuclear envelope. So the outer layer is connected to the endoplasmic reticulum, communication with the cytoplasm of the cell, exchange of the large molecules, that is the protein and the RNA, as well as it carried between the nucleus as well as the cytoplasm happens at this particular juncture. Nucleolus, the largest structure present inside the boundaries of the nucleus. Dark staining zone in the center of the nucleus, where intensive synthesis of the ribosomal RNA takes place, which is very important. Ribosomal RNA, it is going to play an important role further in the transcription as well as in the translation process, where it ultimately leads to the synthesis of the proteins. So the main components are the ribonucleic acid, that is the RNA, and the deoxyribonucleic acid, that is the DNA and the proteins. So it is a place, we can say, very important, very intensive, where intensive synthesis of the rRNA takes place, that is in the nucleolus. Nucleoplasm. It is a jelly-like, made mostly of water, right? So it is a jelly-like matrix which is present within the nucleus. All the materials, they float inside. It helps the nucleus keep its shape and serves as the median for the transportation of important molecules within the nucleus. So it is a jelly-like matrix within the nucleus. All the other materials float inside, helps the nucleus keep its shape and serves as the median for the transportation of important molecules within the nucleus. Chromatin and chromosomes. So chromosomes contain DNA in a condensed form attached to a histone protein, and chromatin is comprised of DNA, and there are two types based on the function. There are two types which are based on the function. That is, one is called as a heterochromatin and other is called as a eurochromatin. Heterochromatin, they are highly condensed, transcriptionally inactive, mostly located adjacent to the nuclear membrane. Whereas the Eurochromatin, they are delicate, they are less condensed, organization of chromatin located in a transcribing cell. They are delicate, less condensed, organized of the chromatin and located in the transcribing cell. So we said that this chromatin, they are of two types. One is a heterochromatin, other is a Eurochromatin. One is highly condensed and transcriptionally inactive, mostly located near the adjacent to the nuclear membrane, that is your heterochromatin. And second is a Eurochromatin, which is delicate, less condensed, organization of the chromatin locating in a transcribing cell. So what is the function? The nucleus is often compared to the command center as it controls all the function of the cells. First of all, it is important in regulating the actions of the cell, plays an important part in creating the cell's protein, and thirdly, it is an important process dealing with DNA and other genetic molecules. We have always been saying that the nucleus, it is only said to be a repository of genetic information. No doubt it is considered as a repository of the genetic information, but at the same time it is functioning as a command center. It functions the regulation of the actions of the various cells, plays important part in creating the cell's proteins, and further it is important in the process which is dealing with the DNA and other genetic molecules. Then comes another very important organelle, that is the ribosomes. The ribosomes are the organelles wherein the synthesis of the protein occurs. These protein synthesis occurs, they are very, very tiny organelles which are present within the cell, whether a prokaryotic cell or a eukaryotic cell. They have ribosomes, and these tiny organelles which are present, they are called as the ribosomes. So ribosomes, they can be found alone in the cytoplasm in a groups which are called as polyribosomes, or they are attached to the endoplasmic reticulum. So when we are saying what are ribosomes, they are the synthesis, they are the sites for the synthesis of the proteins. Now they can be found either alone in the cytoplasm in a groups called as polyribosomes, or they are attached to the endoplasmic reticulum. So when we are talking about the endomembrane system, the endomembrane system consists of the nuclear envelope, endoplasmic reticulum, Golgi apparatus, and the vesicles. When we were talking about the proteins or the synthesis of the proteins by the ribosomes, once the protein is being synthesized by the ribosomes, they have to be targeted towards a specific site. They have to be targeted towards a specific sites, either by the means of various transport mechanisms which are present within the cell. They may be a gated transport, they may be a transmembrane transport, or they may be a vesicular transport. So by the means of three different mechanisms of the transport, the proteins which have been synthesized, they are targeted and translocated to their site of action. Maybe a membrane bound, maybe within the cell, maybe associated with some other organelle, but they will be transported either with the help of a gated transport, they may be transported with the transmembrane transport, or they may be transported with the help of the vesicular transport mechanism. Now this translocation of protein which is taking place within the cell, it may occur by two mechanisms, whether it is co-translationally or it is a post-translational. So translocation of the protein, it can be done by two mechanisms, that is a co-translational mechanism as well as the post-translational mechanisms. And protein always move through a particular secretory pathway. It is not that haphazardly if I want to go from Gandhinagar to Ahmedabad, it is not necessary that I would go in a haphazard manner. I will follow a specific path to go. I will take a specific bus to go. I will take a specific route to go. Similarly, the proteins also move through a pathway which is called as a secretory pathway. Now, the order of this pathway is also very much fixed pathway. That is, from the rough endoplasmic reticulum, it is going to the endoplasmic reticulum to the Golgi apparatus specifically to the Golgi transport vesicles. From Golgi transport vesicles, it reaches to the Golgi cisternae. From Golgi cisternae, it reaches to the secretory transport vehicles, and from the secretory transport vesicles, it reaches to the surface of the cell or to the cell surface. So, the proteins which are made at a particular site in the ribosomes, they are targeted through a particular pathway to their site of action, to their site of residence, you can say, and that is through the secretory pathway in the order of rough endoplasmic reticulum, from endoplasmic reticulum to the Golgi apparatus, that is to the Golgi transport vesicles, from there to the Golgi cisternae, from there to the secretory transport vesicles, or to the transport vesicles, and from there it ultimately reaches to the surface of the cell, that is to the cell surface. Now, further, another important organelle is your endoplasmic reticulum, always emphasized as ER. First discovered in the year 1945 by Albert Claude in Belgium and Keith Porter at the Rockefeller Institute. It was first noticed in the cytoplasm of the chick embryo cell. So on the right side, you are observing a three-dimensional endoplasmic reticulum structure having a nuclear envelope, nucleus, rough endoplasmic reticulum you are seeing, and the smooth endoplasmic reticulum you are seeing. If you see the diagram properly, the rough endoplasmic reticulum, it is having a small dots which are present. That is why it is rough. And whereas the smooth endoplasmic reticulum, you are not able to observe any small dots. But that dots, what are they? We will come to it later on in our class. So what is endoplasmic reticulum? We have a network of railways, we have a network of buses, network of roads. Similarly, in a cell also, the endoplasmic reticulum has a network. So it is a network of folded membranes that will form the channels. So the endoplasmic reticulum is a network of folded membranes that forms the channels. And the endoplasmic reticulum makes proteins and the lipid components. They consist of two things. One is a smooth endoplasmic reticulum and a rough endoplasmic reticulum. And this organelle is very important or responsible for the movement of the protein and the carbohydrates to the other cell organelles, specifically to the Golgi apparatus, to the lysosomes, and other places which need the carbohydrates. So we can say what an important work this endoplasmic reticulum makes. So it makes the protein and lipid components, consist of SER and RER, and responsible for the movement of the proteins and other carbohydrates to the various other organelles like your Golgi apparatus, lysosomes, and other places within the cell when and where there is a need of the carbohydrates. So when we see a rough endoplasmic reticulum, what we see a dotted RER, what we are saying, it is a dotted with the ribosomes, which is why it is called as rough. So why it is called as rough? Because of the presence of the ribosomes, that is a dotted structure which are present on the endoplasmic reticulum. It is called as the rough endoplasmic reticulum. That is why it is called as the rough. The RER which is there, it is also involved with the protein production, protein folding, and quality control, as well as the dispatch of the synthesized protein. So, important functions does lies with the rough endoplasmic reticulum, and thus the rough endoplasmic reticulum which is there, it is involved with the protein production, with the protein folding, with the quality control of the proteins, as well as the dispatch of the proteins to their target sites thereafter. So this RER which is there, it is involved with the synthesis of proteins. They produce and process specific proteins at the ribosomal sites, consist of network like tunnels with tubules, vesicles and cisternae, which is held together by the cytoskeleton of the cell. So this RER, it is important in synthesis of protein, produce specific proteins at the sites of the ribosomal sites, consist of network of tunnels with tubules, with vesicles, with cisternae, which is held together by the cytoskeleton of the cell. Whereas a smooth endoplasmic reticulum which is there, the endoplasmic reticulum, it is a system of membranous channels and sacules. The smooth endoplasmic reticulum lack ribosomes, and it is the site of the synthesis of the phospholipids and packaging of proteins into the vesicles, among other different functions they carry out. So we have seen what the difference lies between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum were always studied with the ribosomes, and they were the site for the synthesis of protein and processing. Whereas the smooth endoplasmic reticulum lacks the ribosomes, and they are the site of the synthesis of phospholipids and the packaging of proteins into the vesicles, among the different other functions they are carrying out. This is the structure of the endoplasmic reticulum. You are seeing the carbohydrate chain, you are seeing the polypeptide glycoprotein, vesicle formation and the transport vehicles. Then comes another important organelle called as the Golgi apparatus. Now this Golgi apparatus, it consists of a stack of curved sacules. What we said that the endoplasmic reticulum also have the presence of the sacules. Similarly, the Golgi apparatus which is there in the eukaryotic cell, they consist of a stack of curved sacules. Now what is the Golgi apparatus doing? What does the Golgi apparatus do? The Golgi apparatus which is there, it receives the protein and also the lipid-filled vesicles from the endoplasmic reticulum. They package, they process and distribute them within the cell. Now, when we are talking that the Golgi apparatus, they receive protein, they are also filled with lipid-filled vesicles from the endoplasmic reticulum. They package, they process and distribute them within the cell. That means they do work like a post office. Post office also receives letters from different places. It sets, it distributes, it makes it different compartmentalized according to the pin codes, and then it is sent to the different places. Similarly, same work is done by the Golgi apparatus within the eukaryotic cells. That is, it carries out the distribution, packages, processes and distribution within the cell. And this organelles is also supposed to be involved in certain secretions. See, if you see the Golgi apparatus structure in this particular picture, that is, the substance is taken. If you are looking at the substance is taken into the cell by the vesicle formation, secretory vesicle discharges a product at the plasma membrane, Golgi apparatus is there. You are seeing the sacules which are being formed in the form of the curved tunnels. If you are seeing at the bottom on the right, the electron micrograph is shown. The transport vesicles move from the smooth ER to the Golgi apparatus. Lysosome combines with the new vesicles and the substance is said to be digested. This particular apparatus, Golgi apparatus, it was discovered by Camillo Golgi in 1897. It was discovered by Camillo Golgi in 1897. It has a series of 5 to 8 flattened membrane bound cisternae located very close to the nucleus, has got two important phases. One is a cis phase, which is said to be a forming phase, and other is a trans phase, which is said to be a maturing phase. Often considered the distribution and shipping department of the cell's chemical products. Modification of chemicals occurs in Golgi apparatus. Substances enter from the ER, from cis phase and exit from the trans phase. N-linked oligosaccharide chains on proteins are altered, and some proteins undergo O-linked glycosylations. Along with them, there is the presence of lysosomes and vacuoles. Lysosomes are those organelles, those vesicles, if we say, they are produced by the Golgi apparatus. Now this Golgi apparatus, these lysosomes, they contain hydrolytic enzymes and are involved in intracellular digestion. And one should know this lysosome is a house of almost 40 different hydrolytic enzymes are present in the lysosomes. Vacuoles, they are large vacuoles, and vesicles, they are small. They are membranous sacs which are present in the cell that store the various substances. What I said earlier, they contain 40 different hydrolytic enzymes, which includes their proteases, nucleases, glycosidases, lipases, phosphatases and sulfatases. And these all are the acid hydrolases, and they require a pH of 5, cannot function within the cell at a pH of 7.2. Peroxisomes. These peroxisomes, they are the vesicles which contain enzyme. The enzymes in these organelles, they use up oxygen and produce hydrogen peroxide. So that means they are catalyzing, they are converting, they are using up the oxygen and they produce the H2O2. These peroxisomes, they are highly abundant in liver, where they produce bile salts and cholesterol, and they break down the fats. That means they are the sites where you can say the breakdown of the fats occur. They are abundant. Liver is an organelle where you are seeing large amount of presence of peroxisomes, where in liver the bile salts are produced, cholesterol is there, and at the same time the breakdown of the fats also occurs. It is a major oxidative reaction center for the oxidation of the fatty acids, and that too by the process of the beta oxidation pathway. That is, by the beta oxidation, the breakdown of the fatty acids occurs in the peroxisomes. Now these peroxisomes, they were discovered by Christian D. Duve in 1965. They are very small organelles ranging from 0.2 to 1 micron in diameter. And these proteins required for peroxisomes formation, they are called as peroxins. So the proteins which are required for the formation of the peroxisomes, they are called as peroxins. So it is important to know that how the proteins, which proteins or what these proteins are called, which are required for the formation of the peroxisomes. Now one could have understood the importance of earlier discussed different types of the cell organelles. And this peroxisomes again is a very important organelle which is carrying out the beta oxidation pathway of the fatty acids. Very important thing with respect to this particular peroxisomes is an important disease, Swelger syndrome. It is characterized by the reduction or the absence of the peroxisomes due to the defect in importing proteins into the peroxisomes. So important inherited disease. This is an important inherited human disease that is called as Zellweger syndrome. It is characterized by the reduction or the absence of the peroxisomes due to the defect in importing the proteins into the peroxisomes. If this occurs, then such type of inherited diseases can occur because of the reduction or the absence of the peroxisomes within the cell. We come to a very, very important organelles that are the energy related organelles. If we talk about the energy related organelles, there are two energy related organelles of the eukaryotic cells. One, if we talk about the plant cells and the animal cells. If you talk about the eukaryotes in general, they are the chloroplasts and the mitochondria. Both organelles which are there, they house energy in the form of ATP. So ATP is a very important molecule which is required for functioning of the cell. It is an energy which is required for cell to carry out different reactions, different functions of the different organelles. For that, every cell or its organelles do require the presence of the ATP, or the enzymes also require the ATP for their work. So chloroplast, it is bounded by two membrane enclosing a fluid-filled stroma which contains enzymes. Membrane inside the stroma, they are organized into thylakoids, and thylakoids house the chlorophyll, and chlorophyll is important, very important for the synthesis of the food for the plants. So what does the chlorophyll do? They absorb solar energy and carbohydrates are made in the stroma. So they are absorbing this chlorophyll molecule which is there, they absorb the solar energy and carbohydrates are made in the stroma. This is the structure of the chloroplast. You are seeing the overlapping thylakoids, independent thylakoids, and the same double membrane. There is a presence of two membranes in the chloroplast. One is an inner membrane and other is a outer membrane. On the left side you are observing the electron micrograph of the chloroplast structure. There also you are able to see the granum, and on the right hand there is a schematic diagram where also you are able to see the granum, independent thylakoids, outer membrane, the inner membrane, as well as the overlapping thylakoids. Mitochondria. They are found in both plant as well as the animal cells. Mitochondria, they are bounded by a double membrane surrounding the fluid-filled matrix. The inner membrane of the mitochondria are cristae. The inner membrane of the mitochondria, they are said to be cristae. The matrix contains enzymes that break down carbohydrates, and the cristae house protein complexes that produce ATP. So what we said in the earlier slide is that the mitochondria, they are bounded by double membranes surrounding fluid-filled matrix. And this matrix, the inner membrane of this mitochondria, they are cristae, and the matrix contains the enzymes which will break down the carbohydrates, and the cristae does house the protein complexes that produces the ATP molecules. This is the structure of the mitochondrion structure, having the presence of the matrix, cristae, outer membrane, inner membrane, that is the double membrane. It is also a double membranous structure, just like the chloroplast. It is also a double membranous structure, further having the presence of the cristae, what we said in that earlier matter, and the presence of the matrix is also observed in this particular thing. Then comes cytoskeleton. Cytoskeleton is very, very important again in the eukaryotic cell. Why so? Because it provides the structural support to the cell. The cytoskeleton also functions in the motility of the cell as well as during its regulation. So providing the structural support to the cell, provides the functions in it functions in the motility of the organisms, motility of the cell, as well as in the regulation of the cell. So when we talk about the structural support, we talk about its mechanical support. So what does it do? It carries out, it maintains the shape. Fibers which are there, they act like a geodesic dome to stabilize and balance the opposing forces, provides anchorage for the organelles. Dynamic, dismantles in one spot and reassembles in another to change the shape of the cells. Again, important thing, they are dynamic, dismantling in one spot and reassembling at the other to change the shape of the cell. So the eukaryotic cytoskeleton which is there, it is a network again of filaments and tubules which extend from the nucleus to the plasma membrane. So the eukaryotic cytoskeleton, it is a network of the filaments and tubules that extends from the nucleus to the plasma membrane. The cytoskeleton which is there, it contains three types of elements which are responsible for the shape of the cell. They are for the shape of the cell, for the movement within the cell, and for the movement of the cell. And what are these three types of elements which are important in the cytoskeleton? They are actin filaments, they are microtubules, and they are the intermediate filaments. So these three important parts, three different elements, they are responsible for the cell shape, for the movement of the cell within the cell, as well as and movement of the cell. That is the actin filaments, microtubules, and the intermediate filaments. Actin filaments which are there, they occur in bundles or mesh-like networks. Actin filaments, they play a structural role in the intestinal microvilli and also interact with the motor molecules such as your myosin. Now furthermore, going into the details of the cytoskeleton and thereafter the discussion of the questions, I will take this remaining part of the cytoskeleton and an overview comparison of, in a brief lookout of the eukaryotic as well as prokaryotic cells and difference between the microtubules, actin filaments and the intermediate filaments in the coming session, which is just after a few minutes.
Vande Gujarat.